so we're fine.
All right.
introduce Professor Bettina Winckler. She's a professor at the Department of Neuroscience and Cell Biology at the School of Medicine of the University of Virginia. And she did her PhD at MIT. And then after, she did 2 postdoc experiences, one at Columbia University and the other one at New York,
Yale.
NYU?
At Yale.
Yale. Okay. I forgot my paper.
We're making it a question-answer session.
Yeah. And she is, her interest is understanding how neurons work, particularly how proteins are trafficking by different organelles in neurons, more specifically working with endosomes. And her talk actually is related with proteostasis. So proteostasis is the synthesis and turnover equilibrium, I guess, of proteins in dendrites. And it's my pleasure to have her here. And please welcome her for this talk.
Well, thank you so much. Thank you for the invitation. It's been a pleasure being here. I've never been down to Blacksburg, even though it's really not that far. So, and it was a beautiful ride, except for the snow in the middle. But other than that, it was a beautiful ride. And I had a wonderful morning talking to everybody. So I'm looking forward to the rest of my day. So thank you so much for having me. So yes, this is the title of my talk. You can see here live imaging of endosomes, and I just want you to have that sense that they're super dynamic, they're always on the move, and we're trying to understand where they're going and why and what they're doing and how they're regulated. But I'm actually going to tell you 2 stories, 2 short stories. One is on this problem of how dendritic membrane proteins get degraded, and we are particularly interested in a set of short-lived proteins I will tell you about. But this story is published, and I just felt badly bringing only published data to you. So I also want to talk to you about a second story at the end that is a very strange story about an unconventional intermediate filament protein called nestin. And so I hope you're kind of like my tryout audience. Okay, so I'd love to hear what you think of it. So I'm going to quickly introduce you to the endosomal system in case you don't think about endosomes as much as I do. So I'm calling this general endosomology. So basically, endocytosis is a way to provide an entry point for all kinds of extracellular molecules. They can be nutrients, they can be growth factors, they can be anything really that's swimming around on the outside. In order to enter the cells, includes pathogens, also enter cells through endocytosis. So it's a really important pathway, both for maintaining normal cellular functions and also in some pathological processes. Then the flip side of that is, of course, that you can remove surface components by endocytosing a receptor. And so if you work on a receptor, you're basically always encountering endocytic processes and endosomal flux because no receptor is on the membrane forever, right? And so the cell really has a wonderful system of changing who is on the surface, where they are, and for how long by using endocytosis. And so I think of the endosomal system and endocytosis as being this wonderfully precise way of spatially and temporally regulating the receptor levels of a cell, any cell, including neurons. Neurons are cells. So some of the features of endocytosis that make it such a powerful way to regulate surface levels that it's really fast. So a cell can remove all of a receptor in 20 minutes. Okay? It's spatially precise, so it can happen in one location, but not 5 microns over. It's regulated, doesn't have to happen all the time, right? And it's receptor specific, so you can have one receptor that is endocytosed and a receptor sitting right next to it that is not. Okay? So it's really an exquisite way of regulating who's on the surface and where. So what happens after endocytosis? Just to point out, because I just skipped over this, is that neurons obviously have endosomes just like all other cells do. And regulating endocytosis, endosomal trafficking is really important for a lot of really fundamental neuronal things during development. Axon outgrowth and guidance depend greatly on surface levels of receptors and how on regulating that level. Neurotrophic signaling is one of my favorite examples of how endocytosis is important to sculpt the nervous system during development, synaptogenesis, and then during neuronal function in the adult. Synaptic function is also really critically regulated by endocytosis and recycling of receptors. So without endocytosis and endosomes, we would not exist, that's for sure. So what happens after endocytosis? And so this is my little diagram of what the endosome can do for you in case you don't think about endosomes. So here's your favorite receptor binding your favorite ligand, and it will signal from the plasma membrane and do all kinds of magic things. And then this ligand receptor complex can be endocytosed into an endosome. And now, since it's off the surface, you have downregulated surface receptor levels. And most of the time, people think of this pathway as being one that ultimately ends up degrading that receptor, and that would terminate the signal. Alternatively, the receptor can actually recycle back to the membrane and reappear, so that would restore responsiveness to a cue. And that can be timed, so the cell can do it quickly or not. The cell can also recycle this receptor to the same place where it endocytosed, or it can take it to a totally new place, okay, and exocytose it there. So you can really just do anything you can think of. Endosomes can probably do it. That's a slight exaggeration. The other thing that we became interested in is how do you maintain a healthy proteome, which means what do you do with proteins that have stopped working well? So you might have some kind of old protein, and there's a separate pathway that gets rid of misfolded or unfolded proteins in parts of the proteasome, which I didn't draw here, or through autophagosomes that would engulf pieces of cytoplasm, and they would fuse with the lysosome and be degraded there as well. So this is really important, and when your proteostasis is working well, this is what we're looking at. We recycle and we trash just the right amounts. But if there's endolysosomal dysfunction, this is what it looks like. And nobody wants to live next to that guy, right? So that's a lot of problem. And so there are all kinds of diseases that are linked to endolysosomal dysfunction, including a lot of neurodegenerative diseases that you might have heard of, where toxic aggregates can accumulate, and clearance of them is impaired, lysosomal storage diseases. And then in aging, all those systems actually slow down, and we start to accumulate more and more trash in our neurons. We got into this question because we had a massive failure. So this is a story of making lemonade, okay? Or a pivot to what we were trying to do. But we started this whole project by being interested in a set of very small transmembrane proteins called neuron-enriched endosomal protein of 21 kilodaltons. So this is a transmembrane protein that's truly tiny. And a related family member that's only 19 kilodaltons, having the not exciting name P19. And they're both now been renamed to NSG1 and NSG2, which tells you actually pretty much nothing except that they're expressed in the nervous system, because this stands for neural specific gene 1 and neural specific gene 2. Okay, so what is known, cell biologically speaking, about these small transmembrane proteins? And it's really not very much. We stumbled into them, and I won't tell you that story, but we know that they can regulate the decision to recycle or degrade. And this is a massive decision point in the endosome, whether you want to recycle or degrade a protein. And NSG1 in particular seemed to be sitting at that decision point for a multitude of important receptors and neurons, including glutamate receptors, which everybody loves, beta APP, which everybody loves who writes grants, because you can say you have an Alzheimer's link now. And we were working on L1, exoryl cell adhesion molecules, and we realized that this little protein was kind of misguiding it, mistargeting it. So what is this protein? Where is it? It's a transmembrane protein. So if you stain for it here, this is a soma, and this is a dendrite from a cultured neuron. You can see all these dots. These are endosomes and the dendrites. EA1 is the most widely used marker for a subset of endosomes called the early endosome. And you can see there are a lot of early endosomes. And they're not like super together, right? Like the red and green are kind of next to each other. So we're trying to figure out, like, where are these proteins? Because presumably whatever they're doing, we ought to know where they are. Maybe we can find out what they're doing. So let me quickly take you through this slide. This is like the markers that people use to distinguish different kinds of endosomes from each other. And there are these different flavors. So here's a receptor coming into the cell by endocytosis. And most of the cargoes that come in kind of fuse into a single place called the early endosome, which the markers are usually used as EA1, which I'm showing you here, or a Rab protein called Rab5. So Rabs are widely distributed through different membranes in cells and recruit specifically with different subsets of membranes in And that is guided by phosphoinositides, which Daniel knows all about, the complications of how that's regulated. But phosphoinositide is really important for recruiting different RABs. So from the early endosome, you can recycle back to the plasma membrane, or you can go to a deeper endosome called the recycling endosome that is marked by RAB 11, so a different RAB, and then recycle to the membrane from there. Or you make the choice to degrade, which is that RAB 5 falls off, RAB 7 jumps on, and then what we now call the late endosome will target you to degradation with a lysosome. And so RAB 7 is the marker for the late endosome, and it also often uses a marker LAMP1, which is lysosomal associated membrane protein 1. And so RAB 7 and LAMP1 are usually used interchangeably to mark the late endosome and lysosome. So this is what we have, and so we said, okay, can we find out where our little protein is in these?
All these endosomes, they emerge from the ER, from the Golgi? Or
The endosomes all emerge, basically, they're maturing from the endocytic pathway, right? But there is...
Where are they originated? Where are they originated, the endosomes?
They're being made by fusing of endocytic vesicles with each other into an early endosome. So you can shut the pathway off, and almost the entire endocytic pathway will disappear. And then if you allow endocytosis, it will reform. Yeah, so they do talk to the ER a lot as well, but not really emerging from it in that way. Yeah. Okay, so where are these proteins? So we carried out this extensive localization study where we did quadruple stainings against endogenous proteins. We think it's really important to look at the endogenous proteins, because if you overexpress, you don't even really can be sure that that's correct. And it's quite a challenge to do 4 colors at once. I don't know if you ever tried to do that, but it was quite a task. But we always use map2, which is here in blue, so that we know that we're looking at dendrite, not an axon. And then we use 3 endogenous markers. So either EA1 to mark the early endosome, RAB 11 for the recycling endosome, RAB 7 as a marker for the late endosome, and then LAMP1 for the late endosome / lysosome. And then we're looking at our little guys. And so just to show you an example of what that looks like, is a little fainter than on my computer, but I hope you can see that there's some red dots here. So this is a dendrite I kind of outlined with this zigzaggy line. And NSG2 in this case is in green. The early endosome is in blue, and the recycling endosome is in red. And you can see that the green and red are really not together, and that there are green dots that are not with blue and some green dots that are with blue. So we count that, and we basically can determine that our little protein is really in multiple compartments, about 30% with the early endosome, almost none, very low with the recycling endosome, very high with the late endosome, RAB 7, but stunningly low with the other marker for the late endosome, or lysosome, which is LAMP1. So that discrepancy was a little puzzling to us, and so we decided, let's figure out what is the half-life. Maybe it's degrading, so we don't really see it accumulate in the lysosome all that well. So we did some what we call biochemistry, but it's not, serious biochemistry. We make cultures, and then we add a drug called cyclohexamide, which inhibits translation. And then at 1, 2, and 4 hours, we scrape the cells, make a lysate, and blot for different antigens, including NSG1 and NSG2 shown here. And you can see that by 2 hours, there really is significant decrease in the levels. So the half-life is about 90 minutes for these little membrane proteins, okay? Here's another receptor, also has a fairly short half-life. If we look at a lot of other proteins and we blotted everything and we had a good antibody against, they're pretty much way more stable. These are all components of either the late endosome or lysosomes. Cathepsins are actually part of the lysosomal proteases, so they are in the lysosome. But they're very stable, so they're not digesting themselves. And then these are some membrane proteins. And you can see that they're also on the order of 4 hours. We don't really see a big decrease. So these are very unusual proteins because they come in by endocytosis. We could show that. They're in the early endosome, late endosome, and boom, they trash themselves in 90 minutes. Okay, so they're just fluxing through the system really quickly. So we're calling them short-lived for that reason. And the reason I'm saying this was our lemonade project is that we made the mouse for NSG1. It's viable and fertile, it's hopping around. To a cell biologist, it looks like a happy mouse. What do I know? And so we started just saying, oh, we can follow degradative flux really easily because we have a protein that trashes itself. And so if we mess up degradative flux, we see massive accumulation of these proteins. So we have a readout for how quickly the cell is fluxing to the lysosome. Okay. So, simple question then. was, okay, we have a protein, I showed you, it's all around in the dendrites, quite far out, but it trashes itself really quickly. So where is that happening? So does the terminal degradation of this short-lived protein take place locally in the dendrites, all along the dendrites, okay? Is that why it's so short-lived? Or basically what we're asking is, where are the lysosomes in the dendrite? Okay, that's the question. They could be in a soma, they could be all along the dendrites. You might have to endocytose into some compartment and then move that compartment to degrade it. So how do cargos get to the lysosome? I already pretty much told you, so I'll just quickly review this, that basically in the early endosome, you make a decision to recycle or degrade by going on to the late endosome, which is the RAP7 LAMP1 positive compartment, and that fuses with a lysosome, which has low pH, and that is where the cathepsins are active. So they're only active at low pH, and so the lysosome is a low pH compartment that has degradative capacity. That's how we're defining it operationally, okay? So in fibroblasts, which is what I'm showing you here, 80 to 90% of RAP7 is with LAMP1. So this compartment, people really use those markers interchangeably all the time. And the rest is probably the RAP7 is kind of on this early, hops onto the early endosome as it transitions to the late endosome. So some of it is kind of on this early endosome. So overall, this is a story of markers, the good, the bad, and the ugly. So you've got to be careful with your markers. So here's an example of this, where we stained MAP2 to see the dendrites, and then we're staining markers for early and late endosomes. And here are the single channels, and hopefully you can see that there's a lot of RAP7 in the soma, but also along the dendrites. EA1 already showed you is in the soma and all along the dendrites. This is NSG2, and again, is in both early late endosomes, as we already showed you, and I showed you it's very dispersed as well. So these compartments are not clumped up at all. They're really dispersed compartments. They're all the way out to the end of the dendrites. So how about a later marker, okay? RAP7, late endosome, but like a true lysosome. So we started also using LAMP1 in conjunction with RAP7, and what you hopefully can see, but it's counted here, is that there's RAP7, and there's still some RAP7, and there's still some RAP7, and you can see a lot of LAMP up here, but then there's fewer and fewer of them. And so when you look at the colocalization, if you count it, close to the soma, or in this first little segment of the dendrite, which we're calling the proximal segment, 0 to 25 microns from the soma, the colocalization is 70 to 80%, which is very similar to what the fibroblast number would be, and that's why people use them interchangeably. But then if you go out 25 microns, it drops to about 50%. If you go out 25 microns, it drops to under 30%. So what we actually see in the dendrite, which is very different from the fibroblast, okay, this is the same. The somatic RAP7 endosomes contain LAMP1. That's just like the fibroblast. But the dendritic RAP7 endosomes frequently don't have LAMP1. And conversely, the dendritic LAMP compartments frequently don't have RAP7. So what are these compartments? It doesn't really fit in this model that the fibroblast people have. So here's a higher resolution confocal image, and LAMP1 is here in red, and MAP2 shows you the soma and the dendrites. And you can see these really large LAMP1 positive compartments, which are mostly in the soma. And they're so large that we can resolve the lumen. So they look like donuts, right? So they're really quite large compartments, and we think that these are the lysosomes. But then we also see very small red dots along the dendrites. I hope you can see them. And they don't resolve as donuts, not because they don't have a lumen, but we just don't have the resolution on this microscope. And so the question is, are these small compartments lysosomes? Are they degradative, right? So we then said, okay, let's look at where are the enzymes. Where's cathepsin? I don't know why people don't stain for cathepsin when they want to find a lysosome, because the antibody is great. It's a great antibody. I don't know why, I always use LAMP1. So you see cathepsin, there's cathepsin here. And if you look at this little blow up of the 3 channels, maybe a little easier to see, this is a big fat dendrite. This is region 1 here, RAP7, yes, LAMP1, yes, cathepsin B, yes. If you go over here, you know, 100 microns out, RAP7, yes, LAMP1, yes, but not a lot, cathepsin, no. And so we quantified this painstakingly, really quite painful to count a lot, but it's important. And we see that there is a gradient for all the markers, that they're less dense, so this is a density measurement, as you go out further from the soma, but that the cathepsins in particular go to almost 0, past like 75, past 50 really, we almost see none, okay? So there are very, very few degradative compartments or cathepsin-containing compartments. And so that suggests that the distal dendrites don't really have a high capacity to degrade membrane proteins. Okay. This is another essay because we like to ask each question with at least a couple of different ways. Because antibodies are antibodies and they can be tricky, and if the compartment is very small, maybe we can't see it. So there are always caveats to concluding something isn't there at all. And I would never actually say it's not there at all, okay. But there's very little, I guess that's how we would put it. So we're now using a different assay for a degradative compartment. And this is an operational definition where we're reading out where there are sites of active degradation, active proteases. So there's a compound called DQBSA, which you can just dump into your culture and the cells will eat it, okay? And it is labeled very highly with a fluorophore, so it self-quenches unless it starts to be digested, in which case it unquenches and it becomes red. So DQBSA turns red in the degradative lysosome, okay? So the experiment is to feed this DQBSA degradation-sensitive BSA with just Alexa 647 BSA that is always fluorescent no matter where it is, okay? And so here's the soma. You see red and blue dots. And you can see that in the soma, most of the blue dots are also red. But as you go out, you see mostly blue, okay? So we know that they're endosomes because they have BSA in them, but they're not red. Okay, so if you do a line scan, just intensity-wise, you see the blue and red near the soma being of almost equal heights. And then you go on and the blue keep there, and we just see these little red blips, right? So there's very little conversion of the DQBSA to red. And so we quantify that. And so in the soma, the ratio of the red to the blue, right, so degradative capacity in that compartment is high. So we're calling this ratio a lysosome, okay? And then we look as you go further and further away from the soma. Initially, you see a really wide range of degradative capacity. So some are highly degradative, so they're lysosomes, and some have almost no degradative capacity. So they might actually be early endosomes because the BSA will go into all the compartments. And then we have this kind of moderately degradative compartments. And then you go further out and you start to completely lose this highly degradative lysosome type compartment. And you do have a few, but not very many, that turn red a little bit. So they're moderately degradative. Okay, so these little blips that I'm showing you here. So our interpretation is that degradative compartments are actually pretty sparse past the first 25 microns of the major dendrites. Well, I didn't show you, but stunningly, there are almost none in the minor dendrites in the culture. So each neuron has one axon and a major dendrite and then a bunch of smaller ones. And it's really the small ones don't really have it. So I'm always showing you the big fat guy. So this is actually, in some ways, not at all surprising. If you really go back and dig in the literature, like from the 60s, 70s, old data, there's old EM data that would kind of identify lysosomes by the morphology in EM, and they would say it's mostly in the soma. It's also true, as a paper came out in the same journal issue with ours that shows that also in axons, distally, there are very few degradative compartments, and as you get closer and closer to the soma, that's where you start seeing them. The reason this became kind of a strange controversy is that people really like local, right? Not just local food, but local translation and local degradation and local transcription, not so much a thing, but So people are really interested and intrigued, especially in the nervous system, because the cells are huge. And so it feels like you need to disperse your activities throughout the dendritic arbor. It's such a big thing, and the axon is even longer. How can you just have this all centralized? It's got to be locally dealt with. And I think for a lot of parts it is true. But there was work recently that came out that reported that there are lysosomes distally in the dendrites. And in fact, they relocated in response to electrical activity into the spine, okay? So there were two papers that said that. And they were using LAMP1 as their marker. So as I showed you, we don't think all the LAMP1 compartments are actually degradative active. Lysosomes, so they could be a different kind of compartment that's responding. It's really unclear right now. But it's also possible that there is local degradation, so that this low degradative capacity that we can see further out, maybe that is biologically relevant. It could have a very important role. It's just that for our cargoes that we were looking at, which is bulk, what I would call bulk cargo like BSA, or these short-lived receptors that don't necessarily have a synaptic function, that they're not really degraded locally very much. So the bulk of it is being degraded in the soma. And this is a very old, very, very, very old culture that sometimes, you forget to clean out the incubator and you go in there and there's like this really nasty dish in the back. And much to your surprise, the cells are alive and actually look pretty robust. And you're like, whoa, I'm going to stain this thing. So that's the experiment. I found an old dish. Let's see what they're doing. And you can see maybe that there are red and blue dots, but the blue dots are pretty much only in the soma. So that's the cathepsin staining. So we don't, even in very old cultures. So it's not like it's just the less mature cultures have this. The older cultures don't do it. And then we lifted ourselves out of the culture dish, which is still a pretty heavy lift for us. But we used a mouse that sparsely expresses GFP. It's called a GFP Thy1 mouse. And so we made these cryosections and stained against LAMP1 and cathepsin B. And the color sense of some of the people in my lab is a little different from mine. So they do the orange-pink thing here. I hope you can see it. But what we did is use software to segment just the GFP. So we are only looking at the neuron and not all these non-neuronal cells that are surrounding it. And then you can see, hopefully, that there is a lot of pink and orange in the soma for sure, so lysosomes. And there's orange along, not as dense as here, but there are LAMP compartments, but there's very few pink. And I put arrows on these tiny little pink dots. So that is, in a real mouse cortex, P28. This is what we can detect with antibodies as cathepsin-containing compartments along dendrites. So the model is as follows. We think of the endosome as being basically a maturing set of compartments. So cargo will come in from the plasma membrane into the early endosome, which is defined by having recruited certain effectors, such as RAP5 and EA1, onto their membranes. So we're calling these the early endosomes. There's more and more. And then at some point, there's a switch to RAP7. RAP5, EA1 fall off. RAP7 becomes recruited. So there's a transition where we still see both markers, but there's relatively low percentage, maybe 25% of endosomes, EA1 endosomes have RAP7. And then you see a population in neurons where we only see RAP7 but no LAMP1. That's really rare in fibroblast. So we're calling them early-late endosomes because they don't have the LAMP1 on it. And we're presuming, although we haven't demonstrated, that LAMP1 will be delivered to these compartments. It's possible that they're completely parallel pathways and they're not maturationally connected to each other. But right now, our model is that they are. So the LAMP1 itself is a membrane protein. So that has to be delivered to this compartment from the Golgi. Okay? It has to find this compartment and be somehow added into it, which is a little surprising maybe. And then we're calling those the late endosomes, and we think that they're pre-degradative because we don't see cathepsins in them. And then cathepsin appears in a compartment that has both LAMP on it and cathepsin, and usually RAP7. And again, cathepsin is a cytosolic, is a protein that is inside the lysosome, so it has to come through the ER and be added into these lysosomes at some point during their maturation. So we call them degradative lysosomes. And just to remind you that in fibroblast, the staining with RAP7 and LAMP1 is virtually identical. Okay, what I'm not going to show you mostly is that RAP7 is actually not just a good marker, but it's a really important protein in the maturation of this endosomal lysosomal system. So we showed that if you express a dominant negative or you knock down RAP7, our favorite dendritic cargo, NSG, is not degraded. And in fact, this is because these pre-degradative late endosomes are no longer transported. So I'm going to show you some of these movies. We make a lot of movies of things. So this is a cherry construct of NSG1. It's co-expressed with wild-type RAP7. If you express the dominant negative RAP7, this movie is running, but you can see they're basically just wiggling around. They're not moving at all. So in the chymograph, it's called a chymograph, instead of being displaced over time, this is time, they're just sitting in the same spot over time. Okay? So RAP7 is really important to make them motile. And we counted that, of course. And control, for instance, transferrin receptor, which is a recycling cargo, so it doesn't enter late endosomes. It doesn't really care about RAP7. So it will just keep moving. And then we had a big surprise, which is something that the reviewers made us do. It took us a long time to do it, but we were actually really grateful once we're done that they made us do it. LAMP1 motility is not affected by RAP7, dominant negative. So that was a big surprise because everybody thinks that LAMP is in that compartment, so it should be affected. But as I showed you, there's a lot of LAMP that's elsewhere, and that motility is independent of RAP7. So that kind of also strengthens the notion that there's a compartment that is LAMP1 positive, but does not somehow is a Rab7 compartment. We don't know. We call it the LAMP1 only compartment. We don't know if they're even endosomes. We don't know anything about this compartment. And the sad thing is that we can only identify it by like quadruple staining, because we have to say, it doesn't have this, and doesn't have this, and does have this. And so it's a bit tricky to work with it.
So I have a good question. Does Rab7 bind somehow to the cytoskeleton at the same time? So kind of
right. So the Rab7, all the Rabs are small GTPases. And they bind either GDP when they're not active, or then it gets exchanged for GTP by the actions of exchange factors called GEFs, and that can be turned over to GDP and inactivated by GAPs. So it's a highly regulated protein in terms of its activity, and when it's active, it binds a whole number of different effectors, including some that provide adapters for motors. So it can hook itself to an effector that can hook it to a motor. That's the thinking, and we're very interested in these adapters now, and we're working on some of them to understand that better, but that's exactly our thinking as well. How is the motor connection made? So this is just to kind of just summarize this part, which is our model for how bulk degradation happens in dendrites, and I do want to emphasize the bulk, because we cannot exclude that there's highly specialized local degradative compartments that might change synaptic composition. So for us, the vast majority of the degradative lysosomes are in the soma and in the proximal 25 microns of the dendrite. Why it stops so abruptly at that point, we don't know. Is there something there? We don't know. We'd be interested in knowing that. We have distinguished early and late endosomes. They are pre-degradative, and they're motile, and they carry to be degraded endocytosed receptors to the soma for degradation. So it's not degraded locally. It has to move to degrade. And it's, in fact, Rab7 that regulates the motility of these pre-degradative late endosomes towards the soma. If you inhibit Rab7, you don't move and you don't degrade. OK, good? Oh, you have a question?
Basic question. I'm pretty ignorant about endosomes. If an endosome is showing Rab5 statement, do you assume that it got endocytosed right around that area of the membrane? Like, because you talked about just
Right. So most endosomes can move a lot. And so I would not presume that it came from near there necessarily. Yeah. OK, so I'm going to totally switch topic, and so I'm putting a big alert, OK? So forget endosomes. We're going to talk about nestin. If you're a neuroscientist, you probably have heard of nestin, because nestin is everybody's favorite marker for neural progenitor cells, OK? So it's always used as a neural stem cell marker. And so I seem to be in the business of trashing people's favorite markers. People hate it. I can tell you that. People hate when you take their marker. OK, so here's just a little timeline of development. Let's say in the cortex, you have stem cells that are proliferative. They express nestin, which is an intermediate filament protein with completely unknown function in the progenitor cell. Nobody really knows anything about what it does. It's just a marker. I had a student once ask me, isn't it just a marker? I was like, what kind of question? What does that even mean? I don't know what that means. Yes, it was put in the genome just so you have a convenient marker. No, it presumably does something. I don't know. What are you asking me? And then these cells will exit the cell cycle, or after division, they might exit the cell cycle, differentiate into neurons, migrate to the right area in the cortex, then send out axons that target the right areas and make synapses and integrate into circuit. And so once they're post-mitotic, new markers will be expressed, including a protein called DCX and beta-3 tubulin, which is this TUDG1 marker. It's very popular for people to use to distinguish a neuron, because it's DCX and TUDG1 positive, from a progenitor, which would be nestin positive. And so my student, Chris Spott, was making cultures from very early mouse cortex and looking at them very young in culture after one day. And he gets these pictures, which are wrong, because the textbook says this doesn't happen. Nestin in a neuron, OK? And so beta-3 tubulin is this neuronal marker. We also can do DCX, and there's nestin, and not just like this weird diffuse thing, but really, really highly concentrated right at the growth cone. Really distinct localization of this protein that is supposed to not be in the neuron at all. So as you can guess, we invited a lot of trouble by deciding we're going to work on neuronally expressed nestin now. So let me tell you a little bit about nestin. It's an intermediate filament, and And if you hear intermediate filaments, most people's eyes like half closed, they like roll back in their heads. You know, this is the boring cytoskeleton, microtubule dynamic, you know, actin is dynamic, intermediate filaments, oh man, that's like the skeleton in cytoskeleton, right? So I used to think that, actually. I used to make fun of my friend who worked on intermediate filaments, and now I'm a huge fan. I think they're completely understudied. We don't know what they're doing for the most part. They're doing a lot more things than we give them credit for. So just to show you quickly, the monomer is not globular, but it's actually an extended molecule. So they make a dimer, and then they make a tetramer that is anti-parallel. So this filament has no polarity. The two ends look the same. So there are no motors on intermediate filaments because it has no polarity. It's a non-polar filament. These tetramers then can associate end-to-end to make these really big intermediate filaments. And a lot of them are super stable. If you think about keratin, right, that is like, you need like 8 molar urea to even solubilize it, right? It's unbelievably stable. So for nestin, don't think keratin, okay? It's completely different. For instance, it's way bigger. So the most intermediate filaments are about 50 kilodaltons. Here's a very typical one, vimentin. So it has a head domain is required for dimerization, the rod domain, which is required for making the tetramer and the dimer, and then a short tail. But nestin is 250 kilodaltons, okay? Nestin is missing most of this head domain, which is absolutely required for polymerization. So it cannot form homopolymers on its own. It needs to copolymerize with somebody else, for instance, vimentin, or a neuronally expressed intermediate filament called alpha-internexin. And then it has this very long tail. I already said that. It's not required for polymerization. And as far as people think about it, it kind of sticks out because it's not really in the dimer. Okay, so people think of the tails of being just like hanging out like a bottle brush from the filament. Okay. So as I already told you, intermediate filaments are very, very boring. I'm kidding, and generally thought to provide kind of structural support for cells, okay? In neurons, for instance, neurofilaments, which is a neuronally expressed intermediate filament subunit, maintains axon caliber, so we know it's quite important for that function. Nestin, on the other hand, is this kind of stem cell expressed protein, has no known roles for assembly, but is actually known to regulate disassembly of vimentin intermediate filaments during mitosis. And this happens because it can bind to some kinases, like this cyclin-dependent kinase called CDC2. And so that leads to hyperphosphorylation of vimentin, and that will disassemble them. So vimentin filaments will disassemble in mitosis, so they are dynamic to that extent. And it's usually used as a marker, in fact, for proliferative cells, like the neuroprogenitors I was telling you about, but it's also highly expressed in a lot of cancers. But there are some very interesting roles that are coming out for nestin as a scaffold for signaling molecules, okay? And this is the way we're starting to think about this now. So the best example is in myoblast, where nestin has been shown to be a modulator of kinase signaling, okay? And the kinase it modulates is called CDK5. This is a kinase only expressed in post-mitotic cells and non-dividing cells, even though it's related by structure to the cyclin-dependent kinases. And brains of mice without CDK5 are pretty messed up. Neurons don't migrate, and they have all kinds of defects. So it's an important molecule in neural development. So it's known that CDK5 needs an activator. So the cyclin-dependent kinases, as the name says, need cyclin to activate it. CDK5 also needs an activator, but it's not a cyclin. It's called P35. So P35 will bind CDK5, and that will make active CDK5. And we know that in neurons, CDK5 activity is downstream of a number of very important guidance cues, including one called SEMA3A. I'm going to tell you a little bit more about that. So what is already known, that nestin binds P35, the activator of CDK5, and so it can scaffold CDK5 onto itself. So this is where Chris was when he saw this, and he decided he's going to go for it, okay? I told him about every 20 minutes that if this didn't come out one particular way, we're going to stop, okay? So, but he would keep delivering, so I said, okay, I'll let him do it. So here's just to show you DCX and beta-3 tubulin, so this is definitely a neuron. Here's, again, this nestin staining at the axonal growth cone. And interestingly, if you look at the dendritic growth cone, there really isn't any accumulation of nestin there. It likes the axonal growth cone. It's not really going to the dendritic growth cone. So it's spatially localized to the distal region of the axons. It also is only transiently expressed, which would be expected because it isn't totally wrong that nestin is mostly associated with early cells, right? So it is going away pretty rapidly. After, in the culture, so in 24 hours, we see about 60% have nestin. And then as you wait, it very rapidly disappears from neurons. Okay, is this like a weird cell culture thing, right? Or do we see it in the brain? So we did a lot of work on this because people really, really, really did not want to believe this. They really did not like it. Okay, so we've done what I think is probably some of the best nestin staining out there at this point. We have stained with every nestin antibody that you can find in any catalog anywhere. Okay, so we have the same staining with all these different ones. We can downregulate it. And I put this here. This was just accepted. And so we have a lot more control. So if you don't believe me, just please look at the paper because we really worked hard to validate this staining. Okay, so here is a cryosection through an early like E16 mouse cortex. Okay. And this is where the axons are running in this red zone where the alpha internexin is. So this is a marker for the axons. And so the way this looks is actually that the axons are running this way, parallel to what's called the ventricular zone, which is a hole in the brain. And then you have the radial glial cells, which are the progenitors, and they are spanning the width of the cortex. Okay? And these are the cells that express very high levels of nestin. That's what everybody always uses to call it a progenitor. So you can see this really bright stuff here. These are the radial glial fibers. So there's a ton of nestin in them, no question. But you can also see the resolution isn't the best here, but this was actually STED image, so it's very nice. We can see a subset of the axons have nestin on them, and we can block it with a peptide that was used to make this antibody, so we can completely block it, not just the bright staining on the radial glial fiber, but also this tangential axon tract staining. And we've done that with a bunch of other antibodies, so we do think this is correct. Does it matter for anything? Maybe it's just left over or something, right? Because the progenitor had so much nestin, and then you divide, and you have a neuron, and maybe there's just some nestin there. It doesn't really do anything. It's just like hanging out for no good reason. So the first thing that Chris saw is that actually axon length is the same between the neurons that don't express nestin in the growth cone, like this one, and that do. Axon length is not different, but growth cone area is pretty different. So without nestin, the growth cone is quite a bit bigger than with it. So could this be important in something? Maybe early axon guidance events, right after the neuron is born, it makes an axon, it needs to guide. Could it be regulated by CDK5 activity? Because that's really the only thing we know about nestin. It binds P35 activator. And so we decided to look at SEMA3A because this is one of the cues upstream of CDK5 activity. So one of the cool things about SEMA3A is that the response to it varies widely in strength and robustness between different cells, populations. And more interestingly, to me anyway, is that it varies within regions of the same cell. So it is a repulsive cue to an axonal growth cone. So the axonal growth cone will turn away from a source of semaphorin, but it is an attractive cue to dendrites. So the dendrite will grow into a source of semaphorin. Same cell, okay? We're talking about the same cell. The two ends of the cell will respond completely differently to the same guidance cue, same receptor, okay? There are variations of receptor, mostly allowing for some cells not to respond, but it's known already that some intracellular targets in the cascade can modulate the actual response of the growth cone to this cue. So I'm going to show you what it looks like when you put semaphorin. Uh-oh, I went backwards. Okay. When you put semaphorin onto a growth cone in culture, this is not our picture, but they're dumping a whole bunch of semaphorin, so the growth cone is growing. Now the semaphorin is being added. Watch this thing. Oh, no. Okay, it's a very drastic and very rapid response. Collapsing response, clearly, right? This is an axonal growth cone, and it cannot grow in the presence of semaphorin. So the receptors are known. Some of the cascade downstream is known, kind of. CDK5 is downstream of it. P35 is that activator. And so the way a lot of the CDK5 substrates are actually proteins that either remodel membranes or associate with actin and microtubules. And so when these components are phosphorylated by CDK5, let's say a microtubule-associated protein that's a substrate of CDK5, that phospho species would not be binding microtubules as well and might destabilize it or change its dynamics. The same with a substrate of CDK5 that's an actin component. So you could change actin dynamics. You can also change membrane dynamics. Okay, and all these would together change the way the growth cone is behaving. And so in the brain, this is not what happens. We're not encountering suddenly massive amounts of this Q. But the thinking is really that the side of the growth cone that's nearer the source of semaphorin would have a harder time extending, and so that these filopodia would be weaker and retract more, and these filopodia would start to grow more, and so that the growth cone would end up turning away from the high concentration. Okay? All right. So CDK5 is, in our minds, possibly a critical element. So the question is, what if you add into the system a binding partner of this kinase complex? Would that change any of the cascade downstream? And especially if this modulator is only associated with axonal growth cones, but not with other growth cones, and only transiently during development associated with this. So Chris went ahead and he did experiments where he added semaphorin. So this is just to show you what I already showed you before, which is that the growth cones that have nestin are smaller. Okay? If you add semaphorin into the system, you can see that this growth cone completely collapsed. Okay? This is a growth cone that has nestin in it, but the growth cone that has no nestin survives the semaphorin. This is 1 nanomolar for 5 minutes, so it's a very small amount of semaphorin. And that's shown in the graph. We counted that. So basically, the non-nestin expressing growth cones are much less sensitive to semaphorin than nestin expressing growth cones. So we don't know what else is different between nestin expressing and nestin non-expressing neurons. So we also knocked it down just to link it more closely to that particular protein. So nestin, we can deplete it with siRNA, shown here. And so one thing that immediately became obvious is that as you knock down nestin, the growth cones actually become more elaborate. So that's like the opposite of what I showed you before. So that makes sense. It's kind of nice to see that when it happens. So there's the quantification. So what if you put semaphorin on it? And so again, SI control, growth cone is completely collapsed in semaphorin. And here you can really see that the dendritic growth cones do not collapse. Only the axonal growth cone collapses. And here's the SI nestin downregulated neuron. Axonal growth cone is perfectly happy. Okay, so let's count it here. So they're not only bigger, but they also don't collapse in semaphorin. So how is that working? What could be the molecular machinery? So this is now the unpublished data, because we just published what I showed you so far. Chris decided he's going to just see if he can find any neuronal CDK5 substrate as a binding partner of nestin, because that maybe makes sense, right? So if nestin modulates it, maybe that actually should be binding. So he just does IPs in 293 cells. That guy almost bankrupts me. He's always buying more things. So he bought like a cDNA for every single neuronal substrate he could find. DCX, DCLK1, CRIMP2, tau, drebrin, and FAC are all known to be CDK5 substrates. He expresses them either in the presence of nestin or not. So this is minus plus. I don't know if you can read that. And then this is the IPs. And the only thing that comes down in the presence of nestin is DCX. Okay? And the other ones, we don't really see them come down. So DCX specifically interacts with nestin. I'm going to give you 30 seconds on DCX, because it's actually a well-known protein. It's a microtubule binding protein. It's downstream of some guidance cues like netrin. And luckily for us, it's associated with a human disease where the patients have mental retardation and epilepsy because the brain has very poor cortex formed, and a lot of the axon tracts don't form. So again, for grant writing is a nice thing. So it's a microtubule binding protein that can be phosphorylated by CDK5. We already knew that. We also know that phosphorylated DCX has low affinity for the microtubule, will fall off, and the microtubule will be less stable, so it will start to depolymerize. So the question is, how does nestin modulate any of this, or does it?
Did he detect the interaction with endogenous DCX, not the overexpressed?
Yes, yeah, both directions. I made him do it all directions. Endogenous, both directions, expressed, both directions. Don't come back to me until you have it, or I'm pulling you off. So does nestin affect this? Oh, I want to show you quickly, because it's so we see in the cell, this is endogenous nestin and DCX. This is a microtubule bundle decorated DCX, and you can see the nestin is sitting right on top of it. We don't know if nestin is assembled into intermediate filament or not, or if it's just sitting on the microtubule. So this is the kind of questions that we would like to answer. But the question that we do have the answer for is, does nestin affect CDK5-mediated phosphorylation of DCX? That's really the question. If nestin is present, does that make any difference to whether CDK5 phosphorylates DCX? And so here's an experiment in 293 cells, which luckily for us express CDK5 endogenously, but not P35. So we can put P35 in, yes or no. It's still a triple transfection and then IP, which is pretty tricky. And so then we have an antibody to the CDK5 site on DCX, which is this here right now. So that's basically all I want you to look at. So you can see that we can put P35 in to activate CDK5, and we don't really see an uptick on the phospho site. But if we put Nestin plus P35, we see massive increase in DCX phosphorylation. That is quantified here. And these are some controls. For instance, if you use alpha-internexin instead of Nestin, we don't see this. So it's only Nestin that can do this. Okay. This is my favorite slide. He looked at all the other non-binding substrates for CDK5. And just like you would kind of think, if you're a CDK5 substrate, you put control, you don't see the phospho event. You put the activator of CDK5, you start seeing the phospho event. You put Nestin with P35, don't care. Okay. Here, you don't see it. P35 alone will make it happen. Nestin doesn't really change it. Same here, you see an uptick, and it stays the same. But here's DCX, and you can see that basically, just putting P35, you don't see much stimulation of that phospho event. You need Nestin plus P35 to see it. So the weird thing is that Nestin modulates only a subset of its substrates, of CDK5 substrates, not everyone, okay? And that's so weird to me. That's really, truly weird to me. Because you would think that you would upregulate a kinase activity and then all its substrates would be more phosphorylated, or you would downregulate the kinase and then all of its substrate would be less phosphorylated. But that you can put a modulator into the system that specifically upregulates just one of the substrate pathways. I just think that's amazing. Okay, so here is, and I could stop if it's getting too late, but maybe I can finish it quickly. We're doing now some overexpression experiment. You know, it's nice to do downregulation, it's nice to do overexpression. It gives you some power to mess around with mutants, which we're now doing. So basically, again, we're just looking at growth cone size by counting the filopodia. But you can see here, if this is a control cell just expressing GFP, that's the growth cone. If you express this other intermediate filament called alpha-internexin, growth cones are about the same size. If you overexpress Nestin, the growth cones are smaller. Okay? So that's consistent with what we saw with the SI-Nestin, where the growth cones ended up being bigger. So then the question is, does this have anything to do with DCX? So what Chris did, he grew some DCX knockout neurons, and he did the same experiment. So these are DCX null cultures, okay? And so now you overexpress the internexin, and the growth cone size doesn't change. That's the same as before. You overexpress the Nestin. Now the growth cone size does not change in the absence of DCX in those cells. So in the wild type, overexpressing Nestins will shrink the growth cone. In the DCX nulls, overexpression of Nestin will not shrink the growth cone. So it needs DCX for this effect. And then he did a rescue experiment where he re-expressed DCX of a plasmid with Nestin, and he gets the growth cone to at least partially shrink again. Okay, so this effect of Nestin is via its interaction with DCX. Okay, so we're proposing a novel role for Nestin as a scaffold for DCX and CDK5P35 in neurons. And I'm maybe going to do this really quickly or not at all, because this is a bunch of data that I didn't have time to show you. I showed you this. Nestin does not affect phosphorylation of non-binding substrates, only of DCX, which is a binding substrate. This was known before, and we can repeat it. Nestin does, in fact, bind P35-CDK5. We have shown that Nestin interacts preferentially with a non-phosphorylated form of DCX, which is on the microtubule, and not the phosphorylated form of DCX. I showed you that Nestin promotes phosphorylation of DCX by CDK5. And in fact, we can immunoisolate the entire complex if we use a kinase-dead mutant of CDK5. And so then we can bring down Nestin, CDK5, P35, and DCX all together. And so the idea is that you have Nestin bound to DCX, which is on the microtubule. If this is assembled into a filament or not, we don't know. And then CDK5, P35 will come in and be activated, and that will increase the phosphorylation of DCX, which is sitting right there scaffolded, and that will cause instability of the microtubules. So in the context of semaphorin, we think that Nestin actually modifies the signaling downstream of semaphorin by scaffolding the kinase near its substrate. So in the absence of Nestin, DCX is not phosphorylated terribly well by CDK5, and so we see only a pretty stable microtubule, so semaphorin is not that potent, I guess. But if that growth cone has nestin, it now brings together the kinase right near that substrate. That increases phosphorylation of DCX. It will fall off the microtubule, and they'll be less stable, and therefore the growth cone will retract. So we think of it as a gain control mechanism. It's not yes, no. It's not required, but it will gain control the signaling output from the system. Okay. This is the lab. This is Chris. So he's done all the nestin DCX work. This is Chanshu, who is doing all the lysosome stuff together with these two people. Laura does a lot of the live imaging. Lloyd does all the counting, which is really valuable thing. This is Kelly Barford, who I just wanted to point out. She's no longer in the lab. She worked on signaling endosomes, the TrkA system that Daniel and I were talking about earlier. And she did her undergrad here at Virginia Tech. So go Hokies! All right. So she was a wonderful student. We loved having her, and she's moved on to better things. And these are two of my undergrads. And yeah, I think I'll stop.
Any questions for Bettina?
I have a question about this RAP4 label endosomes. So you showed a common one. So it seems like the RAP7 endosomes kind of going back and forth.
That is true.
A lot of net motion, is that?
So we counted the net displacement, and it's about 60 retrograde, 40% antegrade. And that pattern that it zigzags, that is very, very typical. And so one of the things that was really weird in dendrites is that microtubules are of mixed polarity, while in the axons, all the microtubules are oriented plus and out. So in the axon, directional motility of vesicles is a really simple problem. If you hook onto a minus end directed motor like dynein, you're going to go back to the soma. If you're going to hook onto a plus end directed motor like a kinesin, you're going to go to the axon terminal. No mystery. It's really simple. In the dendrite, it's really mysterious because microtubules of both polarity are present. So if you hook onto the dynein, how would you know where the soma is? And the bias is very slight. You're completely right about that. Yeah, and that's all I got to say. I have nothing. I know nothing about why. And it seems to be sufficient because we do see net transport, but there's a lot of going the wrong way if you think of it as that.
So can I think of it just the soma because the activities of degradation happens at the soma, so you can think of the soma as like a sink.
For those bulk cargoes, I do think of them as a sink. Yeah, I think so. There's a lot of degradation that's happening there. And sometimes I think about if you think about proteostasis, right, and disturbances to nutrients or something, right, during starvation, this kind of thing, nutrient sensing pathways, they actually a lot of those pathways talk to the lysosome. They talk to the ER, they talk to the Golgi, but they also talk to the lysosome. So that famous kinase mTOR, right, which regulates a lot of these nutrient sensing pathways, is sitting physically on the lysosome. And a lot of the amino acid sensing is carried out at the lysosome because that's where you break everything down into amino acids and they would be transported out into cytoplasm. So that's who knows. The lysosome knows how much free amino acids are being made by degradation. And so I think of it as almost like if the neuron wants to have some sense of its nutrient status, it's going to be way easier to just degrade in one spot. And that's kind of where I'm at. If you're going to have to figure out how much is everyone degrading in all these different places, how am I going to know? So it makes sense to me that bulk of the degradation might be really central. But there could be small or more specific. Yeah, I don't know. Yeah, but that's kind of how I'm thinking about it. I don't know. I have no evidence that nutrient sensing is restricted to the soma, but that's kind of my fantasy.
So right at the endocytosis site, do endocytic vesicles typically have different receptors in endosomes?
So the receptors that are coming in through clathrin-mediated endocytosis, right? Those pits are pretty small. So some people think that only a small number of receptors can enter through a single pit. And those endocytic carriers initially are clathrin-coded. They're going to have to be uncoded. And then they tend to start to fuse with each other, and they fuse with early endosomes, and they become dumped into a larger container. right? But initially, there could be only a very small number of receptors in a single pit. So the question is whether do different cargoes tend to fuse with dissimilar early endosomes, and do you keep different cargoes apart in the system? There's very little evidence for that. If you look at different ones, they all tend to mix into the same early endosome. So the notion in the field, which could be wrong or partly wrong, is that everything goes to the early endosome and there's a big mishmash in there. Yeah.
Actually, I have a question related with that.
Yeah.
You said that the lysosomes in the soma are larger in size than those found in dendrites, and then the capacity of digesting proteins is larger in those nearby the soma.
Right, the large ones, yeah.
Do you think it's possible that fusion of lysosomes can trigger this degradation because of the size? You can have more larger or more, the lysosomes are more prone to be fused, the ones that are in the soma than those that are far from the soma, or vice versa, there are a higher rate of fusion of lysosomes.
So that is a great question, because we don't really know at what point the late endosome will fuse with a lysosome. That should change the size of the lysosome, right? It is possible that the really large things are actually autophagal lysosomes, because the autophagosomes are very large. When they fuse with a lysosome, the autophagosome is 5 times the size of a lysosome, so they fuse with a lysosome. So that is going to be a gigantic structure. So it's possible that most of the really big donuts are actually autophagal lysosomes.
That's what we're thinking of related with the project we were working on.
Yeah.
Membrane curvature plays a role in triggering all this.
Yeah, so that's a pretty much unresolved question, I would say. And when the pH, the pH needs to drop, where does that happen, and how is the pH set? Because the vacuum ATP is a membrane protein, it's going to be there, but it's not always active, so you have to regulate that so that the pH will drop. And then how that is coordinated with fusion, motility, we really don't understand any of that particularly well right now. Yeah.
I have a question about the chymograph that shows the mobility of late endosomes. Could you please bring the slides?
Okay. So the lamp are you talking about the lamp one? Or?
Something very difficult.
Let's see.
Oh, yes. So on the right side, you see that all of the endosomes are present the whole time. And then on the left side, some of the lines disappears and then reappears.
Yeah.
Does it mean that the stability get regulated as well as the mobility?
Right. So that's probably a technical problem on this side, because they are not moving, right? So you just focus on that, you have your focus perfect, and nothing moves, and they're just going to be there. These, because it's a dendrite, when they dive, so it's probably that this one was just diving down. We're not refocusing, right? We're just in one. So that it, sometimes you even have the feeling, oh, it might come back. So it's probably that the vesicle isn't staying in the focal plane. And the reason I'm saying that is that the marker we're watching is a transmembrane protein. So I don't think it has anywhere to go. We can't really go away. So when we have other markers like EA1 or RAP7, which are cytosolic proteins that get recruited to the membrane in a dynamic way, and they can leave. And so we sometimes see these events where it's really bright, and then it fades away, goes away, even on the stationary ones where we usually don't have a focus issue. And so we think that those effectors, we can watch them disappear or reappear on a stationary endosome. It's really difficult to do that when the endosome itself is moving, because it kind of does this, right? And so we just lose the focal plane. So we don't want to conclude anything from that, even though it's a little tempting to do that. But we're like, this is probably just the focal plane of that vesicle.
And on the next slide, on the chemical, you can see that some of those lines are very straight and do not move at all.
Right.
Some of the thinner lines move around. Does it mean that some of the endosomes that are closer to the soma or further to the soma, is there like a relation between distance of the soma to the mobility of the vesicle?
Right. So we have that on our to-do list to actually do a distance thing, figure out if the ones that are closer are behaving differently from the ones that are further away. And I would guess that the answer should be yes, I'm thinking. But we haven't done that analysis. And I think it would be interesting. What we are now working on is triple live imaging, because we're trying to look at 3 things at once. And then we can really call something an early endosome, late endosome, early late endosome, or lysosome, right? And so the markers I showed you, like the DQBSA, there's some markers that are operationally defining the degradative component. And we can live image them. So we can watch the lysosome that has degradative capacity. And we can hopefully see cargo coming down the dendrite and, entering. I don't know. We'll have to see what we can see. But we're trying to do 3 colors at once, live.
Thank you.
Any other questions?
I have a couple of questions about the nesting.
OK.
Yes, so in our lab, we culture mouse neuron stem cell and E16 mouse cortical neurons.
Yeah.
So I'm very interested in the novel role of nesting. The first question is, during the neuron stem cell differentiation,
Yeah.
nesting expression should decrease, right?
So it definitely, so in the neurons, it's way fainter than in the stem cell. The stem cell is really, 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 really bright, right?
Yeah.
So you could take a picture and you wouldn't see the neuronal staining at all, right? But we think it's real. It's fainter, but it's real. Yeah, is that the question? I think it's way brighter. And at some point, maybe the promoter turns off and you'll just turn it over and it will be less and less, for sure. So we haven't answered the question because in some ways I don't honestly care about it that much, is whether nestin is still being actively transcribed in the neuron. So the review asks us that, is it still being transcribed? And it's like, I don't know. I'm not an RNA person, so we'd have to look at transcriptional initiation. So we haven't tested it, although it's something that I'm very interested in doing, especially since you might not have noticed, because I kind of didn't want to draw your attention to it. But when we co-express nestin, DCX and P35 levels both are way higher. So nestin greatly stabilizes those proteins, and we think that complex is more stable. It's possible that nestin is also stabilized by them. And so we're actually pretty interested in what regulates the stability, and this is presumably proteasomal pathways, not lysosomal pathways, but it could be highly regulated. And so we don't know the answer. Other people have measured it in cancer cells. It's pretty short-lived, but we don't know if that's true for us.
Do you think in nesting crosstalks with early DPSs like CD42 or other control?
So basically, there is this 200 kilodalton piece of nestin that we don't know anything about. So we don't know who binds it. It could be, who knows? Yeah, it could be all kinds of things.
IP and who mass spec?
Yeah, we could do it. We should do it. We could do it. Somebody should do it. We should do it. Yeah. Yeah. So this is, I'm basically showing you what we did 2 weeks ago, and we haven't. Well, no. That's the last set. The last set is only counted a couple of times, so we're not finished. We're not ready to send it.
That's why I think you have to share your secrets with Silke, because Silke is our director of the cell biology program for our students.
Okay.
Bettina is the director of the cell developmental biology program at UVA.
Yeah.
I have no secrets. I can.
Any other questions?
And also when you culture the E16 mouse cortical neurons at day 1,
yeah, day 1.
the expression of nesting is pretty high, right?
Is the highest, right? So if you wait, you will see fewer and fewer neurons have it,
Yeah,
and it'll disappear.
When you culture for 7 days, the nesting will decrease.
Is gone. Is going to be gone. Yeah. So already by 72 hours, only 10% of growth cones will have nestin. So it pretty much, the earliest we looked 12. We might have looked at 12 hours, but it's about 2/3 really early, and then it just goes down and down and down, and then it's gone.
Yeah, and when we culture the E16 mouse cortical neuron at day 1, it's almost around, and then start to grow the dendrites, and then that means when the dendrites grow and decrease, the expression of nesting will decrease.
So in the dendrites, we don't see the nestin, right? So even the positive cells, it's only in the axons, really, that we see it consistently. I might have misunderstood that question. What are you getting at? So you're saying the young cells don't have dendrites? Is that what you're saying?
Yeah, my question is that when the cortical neurons grow with more dendrites, the expression of nesting will decrease?
So the older they get, the more will decrease. Are you asking if they have more dendrites? Do they have less nestin?
Yeah.
No. I don't know. That I don't know. That we've never tried to look at that. You think they have more dendrites, they have less nestin in the axon?
I'm just curious.
Yeah, I don't know. We have not done that. Try to look at the number of dendrites. You can ask. We can talk more about that.
Any other questions? Okay, before I forget, just a small token for you
Oh,
here.
thank you.
For
Thank you
coming
so
and
much.